All right, in chapter 4, we're going to be talking obviously about virus replication, as you can see from the title slide here. And so, if a virus is going to be infective, it has to go through its replication cycle. And that, as we're going to see, involves many steps. So, the virus has to be able to get into a cell because it's an obligate parasite. We have to break down the capsid, release the genome, copy that genome, use the genetic information in the genome to make new viral proteins, and then from those proteins assemble new virions, new virus particles, and be able to release those so that they could move on to additional cells. And we consider this replication de novo, unlike in a cell division where you're taking an existing cell, splitting it in half. In this case, you are really building new virus particles from scratch, right? We're having to go from the recipe, from the genome, to make these new virions. And there's really 7 key steps involved in this replication cycle. And we'll go through these one at a time, but they are attachment, so the virus particle actually attaching to the host cell, penetration, which is how does the particle get inside the host cell. Uncoating would be removing that capsid protein to expose the genome. Replication, in this case, is referring specifically to replication of the genetic material, whether it be RNA or DNA. A large portion of this chapter is actually dedicated to different methods of replication for different types of viruses with different genomes. Assembly is assembling the new virus particles. Maturation could vary depending on which virus you're talking about, but that is steps that would be required to make the new virions infectious. And then release is, of course, releasing it from the infected cell. So in this lecture, I'm going to go through those 7 steps, and at the end, I'm going to talk just briefly about virus growth curves. And I'll have this split into a few parts. This lecture split into a few parts. So let's go through this list. We'll start here with attachment. And so again, attachment is referring to that binding of the virus to the surface of the host cell. And so that interaction is usually going to be due to just weak electrostatic forces between proteins on the surface of the virus, either the proteins extending directly from the capsid, or if it's an enveloped virus, extending from the viral envelope. So an interaction between proteins at the virus surface and the surface of the cell. And you see a handful of images here of different known cell surface receptors that viral proteins interact with. Some viruses, famously HIV, for example, will require co-receptors. So for HIV, there's CD4, which is recognized as the main receptor, and then this CCR5, sometimes called CXCR4, is the co-receptor. So I'm not worried for right now about you knowing the names of all of these. When we read our HIV book, this co-receptor ends up playing a pretty important role regarding some functional cures for HIV. So we'll definitely bump into this one again. And these receptors are important, right? This step of attachment is so critical to infection that there's been a lot of interest in how this occurs, right? What are these cell surface receptors? Is there any way to block that attachment process? So, we know a lot about viral receptor proteins, even for newer viruses. So, for example, COVID-19, that's on everybody's mind these days. The cell surface receptor for that is believed to be this protein called ACE2. And you can see in the cartoon here, here's the ACE2 protein in green on the surface of the cell. Here's your SARS-CoV-2, COVID-19 virus. So you have these glycoprotein spikes extending from the viral envelope, and you would get a specific interaction between viral protein and cell surface receptor, right? And that's for every virus that's been studied. This interaction is always specific. And again, right, our main interest in this is, well, maybe there's some way to block that interaction through using a drug, or in this cartoon they're showing an antibody that would bind to that cell surface receptor, might be able to block viral attachment. And so we say this interaction will determine what's called the tropism of the virus, which is basically what types of cells in an organism could be infected, right? Because not every cell, even in an individual, are expressing the same proteins. Like, for example, with COVID, there might be some cells in certain tissues that express ACE2 or express it at higher or lower levels than others. Might make those cells more prone to being infected with that particular virus. And this can really extend to the organismal level. So for example, mice are not normally capable of being infected with poliovirus because it's been shown that they do not contain the poliovirus receptor. They don't express that protein, don't have it in their genome. Researchers created transgenic mice that did express that protein called CD155, and as soon as they expressed that one single protein, all of a sudden mice were able to be infected with polio, and in fact they got symptoms that were very similar to what you would see in humans. Paralysis is primarily the most significant symptom of poliovirus infection. So what that shows is really, right, all the other mechanisms were in place to carry out the rest of the viral replication cycle. Really the limiting factor was just the presence or absence of that one attachment protein. So they are critically important in determining tropism and whether or not individual species might be capable of being infected with a particular virus or not. So following attachment, there has to be penetration. We've discussed the fact that viruses are obligate intracellular parasites, intra meaning they have to be inside a cell. So they've got to be able to penetrate. So that penetration step is just how does the virus cross the plasma membrane? And there's a few different ways this could happen. So it could happen if you're talking about a enveloped virus surrounded by a membrane. It could happen by fusion of the viral envelope with the plasma membrane of the cell. This is what happens with HIV. That's what they're showing in this cartoon here. So you get the viral envelope fusing with the plasma membrane, and then the interior encapsulated genome can enter the plasma membrane. Aside from fusion, it's typically going to be some flavor of endocytosis. So, there's different types of endocytosis, caveolin-mediated, clathrin-mediated, etc. But the general gist is the same, right? You're having an invagination of the plasma membrane that is then engulfing the virion into an vesicle inside the cell. And what they're showing in this cartoon is their sort of storage compartments in the cell, typically an endosome, or if you're talking about caveolin-mediated endocytosis, what's called a cavosome. But these storage compartments where these endocytic vesicles end up delivering these virus particles to. So we'll see endosomes popping up in future chapters of this book quite frequently, and even in some of the subsequent steps in this chapter for that matter. So the virus gets in, either by fusion or endocytosis, and sometimes this fusion step may actually be happening inside the endosome and can be paired with the process of uncoating, which is what we're going to be talking about next. And so an example of this would be influenza virus. So it's an enveloped virus. It gets taken in by endocytosis, and the influenza virus ends up inside that endosome. So we just go back one slide. Right? So imagine your influenza virus is here, now trapped inside the endosome. The influenza virus itself is surrounded by its own envelope, its own membrane. And we will get this fusion between the viral envelope and the endosomal membrane. So if this is your virus down here, you have these proteins extending from the viral envelope, two key proteins involved in this process, HA1 and HA2. And what will happen is once the influenza virus is in the endosome, HA1 will mediate this initial interaction between the virus particle and the endosomal membrane. It then sort of folds away and allows HA2 to interact with the endosomal membrane a little bit more tightly. And you'll have two of these protein structures side by side. And what will happen is once HA2 is interacting with the endosomal membrane, basically creating this bridge between the viral envelope and the endosomal membrane, these proteins will fold over, and the two that are side by side will fold sort of towards each other. And this is going to have the effect of now pulling the endosomal membrane into contact with the viral envelope. So these two will actually physically join together. And because we have two of these doing the same thing side by side, it's going to have the effect of creating a pore between the viral envelope and the endosomal membrane that is now going to allow the contents of the viral envelope, basically the viral genome, to escape from the endosome into the cytoplasm. And we'll come back and see this again later. But this is basically fusion happening here in the endosome instead of at the cell surface. And it's also facilitating uncoding at the same time. So penetration is an important step. It's been very well studied. We know quite a bit about how most medically significant viruses gain entry to the cytoplasm. And you can see a short list here of includes influenza, HIV, HPV, hepatitis. All right, let's talk next about uncoding, which again is the step of actually getting the genome out of the viral capsid so that it could be replicated, so we can make copies of the viral genome, which will then be used as blueprint for making new virus particles. So uncoding can occur in various places. There are some viruses that think poliovirus actually encodes at the plasma membrane. This would be sort of analogous to a bacteriophage, right, which binds to the surface of a bacterium and then injects its genetic material into the cytoplasm of the cell. So that would be an uncoding step. So there are some human viruses that are believed to do something similar. Or it could be a case like influenza virus, where that uncoding is basically happening in the endosome. Or it could be a case where uncoding just happens in the cytoplasm. Here you see a few different variations on the theme of uncoding at the endosome. But one way or another, we need to get the genome out of the capsid into the cytoplasm, or potentially even into the nucleus, right? So a fair number of viruses might need to get their genome into the nucleus so that it could be copied, because that's where a lot of the proteins and enzymes that would be involved with nucleic acid replication would be located. So you do have some viruses that are actually uncoding at the nucleus, or potentially even in the nucleus. So you've got your nucleus here in purple, the nuclear envelope, and then these are nuclear pore structures. So for example, hepatitis B is so small, it actually enters the cell, penetrates, and then goes straight into the nucleus through the nuclear pore, and uncoats there inside the nucleus. That's fairly unusual. More typical would be like influenza, that's basically uncoding in the cytoplasm or in the endosome, and then its genetic material is being transported through the nuclear pore into the nucleus. You do have things like herpes virus, which travels on microtubule tracks basically to the nuclear pores, and it uncoats right there, or very close to the nuclear pore, so that as it uncoats, basically its genetic material is threaded straight into the nucleus. Or you have things like adenovirus that will physically dock tightly with the nuclear pore and uncoat while it's docked, and again threading its genetic material into the nucleus during that uncoating process. But it's all really, variations on that same theme. How do we get the genetic material, DNA or RNA, out of the capsid? All right, I will stop there and pick this up in part 2.